the day 4 today of our FDP program organized by Department of Pharmaceutical Science and Technology, MACOUT. On behalf of Pharmaceutical Science and Technology Department, MACOUT, we heartily welcome Professor Dr. Kunal Rai. Currently, he is the head of the Department of Pharmaceutical Technology, Jadavpur University. Today, we have with us, he's a great academician and scientist at global level who has been universally acknowledged. So, let me introduce Professor Rai with the participants. Professor Kunal Rai is professor and head of the Department of Pharmaceutical Technology, Jadavpur University, Kolkata, India. He has been a recipient of Commonwealth Academic Staff Fellowship, University of Manchester 2007, and Marie Curie International Incoming Fellowship, which is a prestigious fellowship, obviously, from University Manchester 2013, and was a former visiting scientist of Istituto da Ricerca Pharmacology, Mary Negri IRCCS, Milano, Italy. The field of his research interest is quantitative structure-activity relationship, QSR, and molecular modeling with application in drug design, property modeling, and predictive ecotoxicology. Dr. Rai has published more than 300 research articles in referred journal. Current Scopus index, H index is 43, total citation till date 9,562. He has also co-authored 2 QSR-related books with Academic Press and Springer Nature, edited 6 QSR books, Springer Nature, Academic Press, and IGI Global, and published more than 10 book chapters. Professor Rai is the co-editor-in-chief of Molecular Diversity, Springer Nature, and editor-in-chief of International Journal of Quantitative Structure Property Relationship, IGI Global. He serves on the editorial board of several international journals, including European Journal of Medicinal Chemistry, Elsevier, Journal of Molecular Graphics and Modeling, Elsevier, the Computational and Structural Biotechnology Journal, Elsevier, Chemical Biology, Drug Design, Wiley, Expert Opinion on Drug Discovery in PHARMA, Letters in Drug Design and Discovery, Bentham, Current Computer-Aided Drug Design, Bentham. Apart from this, Professor Rai is a regular reviewer of QSR papers in journals like Chemosphere, Elsevier, Journal of Hazardous Materials, Ecotoxicology, Environmental Safety, Elsevier, Journal of Chemical Information and Modeling, ACS, ACS Omega, RSC Advances, Molecular Informatics, Wiley, ACR and QSR in Environmental Research, T&F, etc. Professor Rai has been recipient of several awards, including AICTE Career Award 2003 and 4, DST Fast Track Scheme for Young Scientists 2005, Bioorganic and Medicinal Chemistry Most Cited Paper 2003 to 6, 4 to 7, 6 to 9 awards from Elsevier, the Netherlands. Bioorganic and Medicinal Chemistry Letter Most Cited Paper 2006 to 9 award from Elsevier. The Netherlands, the Professor R.D. Desai, 18th birthday Commonwealth Medal and Prize from Indian Chemical Society, etc. Professor Rai has been a participant in the EU-funded projects, Nano Bridges and IonTox, apart from several international government-funded projects like UGC, AICTE, CSR, ICMR, DBT, DAE. Professor Rai has recently been placed in the list of top 2% science-wide author database of the world rank 81 in the subfield of medicinal and biomolecular chemistry. So, man like him, he has done enormous contribution in the field of pharmaceutical sciences. So, actually, today he will deliver his talk on on quantitative structure-activity relationship, software tools, and methods developed at the Drug Theoretics and Chemoinformatics Laboratory. So, without wasting much time, I would request Professor Rai to start his presentation. Sir, please.
Thank you very much for the introduction and good evening to all. Today, my topic is quantitative structure-activity relationships. I will first discuss the basics of QSR for the beginners in the field, and then I will discuss some tools which have been developed in our laboratory at Jadavpur University for developing and validating QSA models. Can you see my screen?
Yes, it is visible, sir.
Okay. Okay. So QSR is basically an acronym of quantitative structure activity relationship. So this is a statistical approach where biological activity of a set of compounds is correlated with the chemical structure information. Now, chemical structure information represented within a numerical form called descriptors are used to correlate biological activity. Now here, as you know, the chemical structure information is actually graphical information, or you have the pictorial representation of molecular structure. So we have to convert the information encoded within the molecular structure into some numerical form so that we can apply statistical approach. So descriptors serve that purpose, where we can store information about molecular structure in numerical form. And we apply appropriate statistical method to develop the relationship, which may be either regression based or classification based. Now, in some cases, biological activity may be in continuous scale, and in some cases it may be graded in nature, like toxic or non-toxic, or active or inactive, and so on. So if the biological activity is collected in a graded fashion, in that case, we have to go for a classification-based model. Or if you have quantitative continuous biological activity values, then we'll go for regression-based models. But the basic notion of any QSR model is that the biological activity is a function of chemical structure and or property. Or biological activity can be expressed in terms of descriptors. Now you can see this tree structure, phenothiazine drugs. They only differ in the substituent pattern. The first one is clopramazine, which is antipsychotic. The second one is promethazine, which is antihistaminic. And third one is ethopropazine, which is anti-Parkinsonian. So due to small changes in structural pattern, they show completely different pharmacological activity. So there may be changes in the type of activity, there will be change of the duration of activity, and there may be change in the magnitude of the activity. And such changes occur in a systematic fashion, which can be modeled, and which is the basic notion of model. The relationship between molecular structure and changes in biological activity in a quantitative fashion is the center of focus of QSR. Although we are using the term QSR, but it can include the other responses like property and toxicity also. That means instead of biology, we can also model property of chemical compounds or drug structures, and also toxicity of chemicals and drugs. So QSR is a general term which can encompass study of both structure activity and structure property relations in broad sense. And it is an intellectual exercise of assembling, manipulating, and examining data obtained from physical, chemical, biological, and computational experiments, and correlating these to the response using chemometric tools. As I've already mentioned, the basic relationship is like this: model response, which may be either activity or property, is a function of chemical structure and property. QSR is a ligand-based design, so we have to start with molecular structure information, and we have to represent the molecular structure in appropriate form. And there may be different level of theories in which the descriptors can be computed. That depends on the structure representation, whether it's in 2D or 3D level. So once we represent the structural information in appropriate form, then we encode the information in numerical form, which are called descriptors. And then we apply appropriate statistical technique and fitting techniques to derive models, which can then be used for prediction of the target property or activity. So these are some representation of the molecular structure. These are 2D structure representation from which we can gather some information. And then we can get the different descriptor values. As you can see here, this is a matrix, and each column represents one particular descriptor. That the descriptor may be simple information like say log p value or pk value, or maybe information like say number of say double bond, or number of particular type of atom like say carbon atom, or number of rings. So it may be as simple as that, or it may be the higher level of information also, maybe some information obtained from quantum chemical calculations. So whatever information we get about molecular structure or about their properties, we can tabulate them in this fashion, where each column represents one particular descriptor and each row represents one data point or compound. Now, in this way, we can derive so many descriptors. So it is our job to uh, do the data reduction. That is, we have to omit or eliminate descriptors which are not much important for our particular response. So we can do data pre-treatment to reduce the dimension of the descriptor matrix, and then we can apply appropriate feature selection tool to know which features are actually important for our target property or response. Now we can calculate thousands of descriptors. We can derive different features, but all descriptors or all features are not important for our particular activity or toxicity. So we have to extract that particular information, that which descriptors or which features are actually important for our particular response. And then we apply appropriate statistical fitting techniques to derive our regression or classification based model. And in most of the cases, we get an equation. And in case of regression-based equation, an equation may look like this. In the left-hand side, we have the response, and in the right-hand side, we have the different descriptor combination along with their weightage. Now, then this model should be evaluated for its statistical quality, because once you apply any statistical technique, you get some model, but we have to check what is the statistical reliability and robustness of that model. And then we have to apply a process which is called validation. Validation means whether this model will be applicable for new set of data or not. So once the model passes through all these rigorous checking processes, then if we are satisfied with the quality of the model, then we can apply this model for prediction of new set of compounds. We can study the features which have emerged in that particular model or equation. We can interpret the model, and we can use it for mechanistic interpretation of our particular response. Using the developed model, we can also design new compounds and predict the values of the response of those compounds using the derived model. Now, there are some basic steps for development of an QSM model: data preparation, processing, prediction and validation, and interpretation. As I've mentioned, that QSR is a ligand-based approach. We start with the chemical structure and representation. Then we derive the descriptor values, and you prepare the QSR table. And then we apply data pre-treatment to remove the intercorrelated descriptors and the redundant descriptors to reduce the dimensionality of the matrix. And then what we do, we divide the data set in two parts. One part is called training set, and the other part is called test set. The test set compounds are kept aside, and they are never used in the model development. They are unseen to the model. We use only the training set of data. So we use training set compounds for model generation and the learning process. And we apply different feature selection tools, and we can apply different statistical methods for developing either regression-based or classification-based models. And once we develop the model, then we apply different validation strategies to check the reliability of the model for new set of data. Now here comes another important point that is called applicability domain. The model that we have developed may not be applicable for all kind of chemicals. So they will be applicable only to restricted set of chemicals which are similar to the training set of data. Because as QSR is a training process, so it can only respond to the features which has been shown by the training set compounds. So the features which are not present in the training set compounds cannot be represented by the model, and model cannot respond well for those kind of features. So that is a point that one should keep in mind. And the test set compounds which are unseen to the model can be used to validate the model, because we can use the model to predict the response values for the tested compounds. And as we have already the experimental values for the tested compounds separately, so we can then compare the predicted values with the experimental values to check the goodness of predictions. And we have different kind of metrics to know what is the quality of predictions. And finally, we can interpret the results to understand the physiochemical basis of the particular kind of response. And we can use the model as a guide for designing new set of chemicals. Now, QSR has different application areas. It is used in medicinal chemistry for drug design, but apart from that, it can be used in material science for fine-tuning of property, for enhanced operation selectivity and optimized process efficiency. It can be used in predictive toxicology, both for systemic toxicity and ecotoxicity, for risk assessment, hazard identification, hazard categorization, and prioritization of chemicals, which can help to reduce the burden of synthetic program and experimentation. So in QSR, we are actually using knowledge from the limited number of chemicals. So we need to synthesize and test a limited number of chemicals only, and we can use that data to derive the statistical model, which can ultimately be used for prediction of very large number of compounds. And it may so happen that we can apply it for hypothetical compounds or virtual compounds also, the compounds which have never been synthesized and tested. And in that way, when we have predictions for those hypothetical or virtual compounds, and if we see some compounds promising, then we can go for synthesizing and testing for those promising compounds. And in that way, you can reduce the cost of synthesis and the manpower and time involvement in those processes. As you all know that drug development is very costly and time consuming affair. This is a long process. It can take 12 to 15 years, and there are several stages of drug development. It's already known to you for all pharmacy background professionals. So I do not need to repeat this. So as the cost of drug development is very high, so the failure is very costly. So we should increase the probability of success. And if the compound fails, it should fail early in the drug discovery pipeline. And to have the idea about the failure potential of a compound, we can do so early in the stage of drug development by application of some theoretical model. So QSR is basically a filtering tool by which we can know what is the potential of a compound in terms of its activity and also in terms of its pharmacokinetic behavior. There are several examples, successful application of QSR in drug discovery. Here, QSR can be used along with other modeling tools and structure-based design. So some successful examples are tirofiban in thrombosis, imatinib in cancer, raltegravir in AIDS, donepezil in Alzheimer's disease, and so on. As I have already mentioned, that QSR is not only applied in medicinal chemistry, but it is also used in environmental science to predict chemical hazard of industrial chemicals. As you know, there are thousands of industrial chemicals, and new chemicals are coming to the market at a very high rate. So we cannot experimentally determine the effect of these chemicals on different environmental species, because all these chemicals are coming to the environment. So environmental species, the aquatic, different aquatic species and terrestrial species are exposed to these chemicals, and they have the hazardous effects. So there should be some models which can predict the effect of all these chemicals, because there are thousands of endpoints and we do not have sufficient resources to experimentally derive data uh, about the hazardous potential of all such chemicals. In fact, we have data for less than 1% of the compounds about their hazardous potential. So with the ever increasing production of new chemicals and rising cost of experimental testing, different regulatory bodies like US EPA and European Commission's Scientific Committee on Toxicity, Ecotoxicity and Environment recommend QSR for data gap filling. So in cases we do not have experimental values, we can use QSR derived data to bridge the data gaps. So as I've already mentioned, that due to shortage of experimental data, QSR estimates can be used for selection of persistent, bioaccumulative, and toxic, that means PBT substances. QSR can also be used to design greener chemicals, and they can be used to explain the mechanism of toxic action of chemicals. QSR is very important in the context of REACH regulation in the European Union for the bulk export of chemical compounds. The industries have to supply or the toxicity data, and in cases some data are not available, the QSR derived data can be used as a supplement. So QSR is supported by different organizations like European Center for the Validation of Alternative Methods, International Organizations of Medical Sciences, US EPA, and OECD, Organization for Economic Cooperation and Development. There are different endpoints in which QSR can be used for predictive toxicology. For example, genotoxicity, nephrotoxicity, developmental toxicity, dermal toxicity, eye irritation, skin erosion, etc. In material science, it can be used for different kind of materials like biomaterials, fullerenes, catalysts, carbon nanotubes, polymers, etc. So as I've already mentioned about QSR table, where you can, it looks like this, where each column represents a particular descriptor and each row a particular data point. Now the basic concept is that the number of descriptors should actually be low in the final model, but number of molecules should be higher. Because QSR is a statistical approach, so there should be favorable degrees of freedom. So for favorable degrees of freedom, you should have more number of compounds and less number of descriptors. Now, if the model is good enough, then the prediction that you make from your model should be close to the 45-degree angle line when you plot the experimental data against the predicted data, and the degree of scatter should be low. Now, if you see the historical perspective, QSR, the concept actually generated quite long back. So we can consider the Hammett theory for the electronic Hammett constant as a starting point, where mathematical QSR was applied. Before that, there was the concept in the Mayer-Overton for correlating narcotic potency with the olive water partition coefficient. But the truly mathematical treatment was done by Hammett in 1935. And QSR was basically applied in physical organic chemistry instead of biological field to model the ionization constant of substituted benzoic acid derivatives. A latter Hans applied this the Hammett concept in the biological field, and he introduced the hydrophobic substituent constant concept. In the year 1962, Free Wilson developed this mathematical model, the de novo model, and then in 70s, the graph theoretical applications flourished, and we got several topological descriptors. In 80s, the 3D QSR originated through COMFA, and it was then followed by COMSIA, comparative molecular similarity analysis. And then it is growing. And then we have from 90s different virtual screening studies, ligand-based and structure-based approaches, and then multi-dimensional QSR formulation. So QSR can be described in terms of different dimensions, like 0D, 1D, 2D, 3D, 4D, 5D, 6D, 7D. So the 2D is actually based on the graph theoretical representation, and below 2D, the substructural and like say some property based can be described in terms of 0D and 1D. The 3D is spatial geometry derived. In case of 4D, there is conformational aspect. In case of 5D, one has to use the induced fit parameter. In case of 6D, solvation conditions are included, and 7D is the target-based receptor ligand interaction descriptors are included. QSR can be used in association with other related techniques like pharmacophore, then different structure-based design, like including docking, different cheminformatic approaches, and database mining. As I've already mentioned, QSR is mainly of two types, regression-based and classification-based. In case of regression-based, it is used when you have the biological activity values or response values in continuous scale. And when you have graded data, one can use the classification-based approach. The simplest regression-based approach is method of least squares, and the more robust approach is partial least squares. In classification-based approach, one can use discriminant analysis or cluster analysis. Machine learning approaches are very popular nowadays, and they can again be regression-based and classification-based. Some examples are artificial neural network, support vector machine, random forest, and so on. Now, there are some requirements of biological activity data for doing QSR, especially for classical type of QSR. The response value should be the dose fixed response time, like IC50, EC50, etc., because they are multiple point assay and statistically more meaningful. The concentration should be measured in a molar unit, and the molar concentration should be converted to a log basis. That means we actually take log 1 by C or PC as our response, because a compound having, say, IC50 value, lower IC50 value is actually more active. So it is inverse relationship between activity and concentration. And when we are using dose for fixed response, so response is fixed. So we are changing the concentration or dose only. And there should be a span of 4 to 5 log units. So usually for biological QSR, we have to convert the concentration, the molar concentration values in a log unit because of two reasons. If you have a dose response curve that is parabolic in nature, but when you use log dose response curve, that is sigmoidal in nature, and the middle portion of the sigmoidal curve is linear, which is easier to model. And the second reason is that we have several metrics for determination of quality of the model. So if you use concentration, that will be a wide range, like say 10,000 or more in a molar scale. So the metric values will be biased towards higher value. So to avoid that, the biological activity values are converted to log scale. And there should be sufficient data points. So number of compounds should be higher and number of descriptors should be lower. So in the simplest case, if we have the multiple linear regression model, it will look like this. So as you can see in the left-hand side, you have the dependent variable or the variable that you are going to model, or the biological activity or property or toxicity that you are modeling. In the right-hand side, you have the independent variables x1, x2, x3, which are actually descriptors. So these descriptors come from the chemical structure or their measured properties. So x1, x2, x3, these values are known to us. And the biological activity values are also determined experimentally for training set compounds. So what we have to determine is the values of these coefficients, A1, A2, A3. These are called regression coefficients. And when we have our model derived, we can use this model for prediction of new set of compounds, because for new set of compounds, we will know this x1, x2, x3 directly from chemical structure. And as we have already determined the values of the coefficients, we can use this model to predict the response values. And then we can plot the observed versus predicted values. And if the model is good enough, we'll see the degree of scatter will be low, or the points will be near this 45-degree angle line. Now, there are several metrics by which we can actually calculate the quality, statistical quality of the model. Now, for the calculated values, if we compare calculated values with the observed ones, as expected, the difference should be small, but there should be a point of reference, how much smaller it should be. So the point of reference is the mean of the training set response. So if we compare the individual observed values with the mean of the training set response, so obviously this difference should be high. So if we see this expression, the denominator should be higher and numerator should be lower, a smaller value. So ideally, these values should be close to 0, and then R square will be close to 1. So that is called determination coefficient, and square root of this is called correlation coefficient. But this doesn't tell anything about the number of descriptors used in the model. So if you go on increasing the number of descriptors, so you will get a value of R square near about 1, but that doesn't mean that you have a good model. So you have to penalize the model for the number of descriptors. So you can do so by using the term number of descriptors here. So that is called explained variance. So where we are penalizing the model based on the number of descriptors. Then comes variance ratio, which gives us an idea about the stability of the regression coefficients and the significance of the determination coefficients. So the F is the variance ratio. It has 2 degrees of freedom. And then standard error of estimate, it should be a low value for a good model. Now, for classification of this model, we have different kind of metrics like sensitivity, specificity, accuracy, and precision, which are derived based on true positive, true negative, false positive, false negative values. Now, OECD has given several guidelines for development and validation of QSR models. And what are these guidelines? Guidelines are there should be a defined endpoint, and when we are using a data set for development of QSR model, we should take care that the biological activity values have been determined using the same experimental protocol for all the compounds. We should use an unambiguous algorithm for development of the model. There should be a defined domain of applicability. When we apply the model for new set of data, one should check that model is applicable for the new set of data. There should be appropriate measures of goodness of fit, robustness, and predictivity. And there should be a mechanistic interpretation of the model. So as I mentioned, the QSR models are developed based on QSR OECD principles. So we can show that which steps actually are complying with which principles. The starting point, the defined endpoint is with the OECD principle 1. Then unambiguous algorithm, that means molecular structure representation, descriptor calculation, division into training and test set, then the application of appropriate statistical method for feature selection and model generation. This comes under OECD principle 2. Then OECD principle 4 states about the different statistical metrics to know the statistical quality of the model and validation pattern. OECD principle 3 states about the applicability domain, and OECD principle 5 is for mechanistic interpretation of the model. So one should also consider several other points during model generation. For example, data homogeneity. Now, if we are actually clubbing two different data sets from two different laboratories, so it may so happen that the same compound may have two different activity values determined at two different labs, and that is due to the inter-laboratory variations or due to some changes in the experimental conditions and so on. So one should take care about this, and one cannot mix such two data sets. Then inappropriate endpoint data, I've already mentioned that there are some requirements for the biological activity endpoint values for use in the QSR. Then collinear descriptors, the descriptors which are intercorrelated should be avoided in general unless you're using some robust method for model generation. The incomprehensible descriptors, the descriptors without any meaning or which have some mathematical background only without any physiochemical significance should be avoided. Error in descriptive values. The error can originate from several sources. For example, say you are using a particular compound with a particular tautomeric form, and so the descriptive value should be something. But if in some other compound for the same feature, if you are using this other tautomeric form, the descriptive value should be completely different. So one cannot use the two different tautomeric form for the same structural feature in the data set. So one should take care about this. In addition, there may be the presence of some structural features which cannot be handled by a particular software. For example, occurrence of some metal or some other structural feature which may not be handled by the software. So one should take care about that also. Then transferability of QSR. The model that one generates should be transferable and reproducible by other group of people also. Then one should take care about the applicability domain, unacknowledged omission of data points. Sometimes the people omit some of the compounds or data points to improve the quality. So the unacknowledged omission should be avoided. Use of inadequate data. For QSR, one should use sufficient number of data for model generation. Replication of compounds in the dataset should be avoided because it may so happen that you are collecting your dataset from several databases and some compounds are present in two different names. Say in some database it is present as aspirin, in some other databases it is present as acetylsalicylic acid. So same compound may be present in two different names. So that should be curated. Narrow range of endpoint values. If you have the quantitative, the biological activity values with range very narrow, such data set may not be suitable for QSR analysis. Then overfitting of data should be avoided by reducing the number of descriptors used. So use of excessive number of descriptors should be avoided. Lack of inadequate statistics for checking the quality of the model is not permissible. Incorrect calculation should be avoided. Lack of descriptor autoscaling. This is sometimes important to understand the relative contribution of different descriptors. Misuse or misinterpretation of statistics should be avoided because usually QSR is done by non-mathematicians. Consideration of distribution of residuals can help us to understand the pattern of error. Inadequate training and test selection, failure to validate QSR properly, and mechanistic interpretation. So these are the points one should consider during development of the model. Now, the most important point about QSR is that the validation. One can develop a model, but one should be sure that it can be applied for new set of data, because ultimately the QSR model will be used for new set of data for which we may not have the experimental data available. So there are several methods to validate a model, like internal, external, and also wide randomization. In internal validation, we use the the same set of compounds in the training set for validation purpose, whereas in case of external validation, we use a completely different set of data. So in case of internal validation, we can omit some of the compounds from the training set, and we can develop the model from the rest of the compounds using the same set of descriptors selected in the original model. And then we can predict the omitted compounds using that developed model. And then we can compare the predicted values with the observed ones. And we can then use some metric like say Q-square, which looks like the determination coefficient R square. Only difference is that we are using here the predicted leave-one or predicted values. So this cross validation can be done either by using leave one out or leave many out methods. Then external validation, in case of external validation, we can use a completely different set of data, or we can use test set compounds, which is originally derived from the data set, but we have kept them aside, and we have not used those data for model generation. And we can use the model for prediction of test set compounds, and then we can compare the predicted values with the observed one, and using appropriate metric like Q-square external F1 or R-square pred to know the quality of prediction. One can also use the Y randomization, where one can be sure whether the model is obtained by chance or not. Here we can randomize the Y column or the observed values, experimental values, and we can redevelop the models, and we can do it several times, say 100 times, and we can see for these randomized Y columns, what sort of models we get. And obviously for the random models, quality should be poor. And then we can use some metric to judge the quality of the model and robustness of the model. Selection of training set is also important, but I don't have so much time to explain all these in details. There are some methods of training set selection, like statistical molecular design, Cohen's self-organizing map, K-means clustering, K-Nastone, etc. So most of which are based on similarity principle. I've already mentioned that applicability domain is very important in application of the model for new set of data. And there are several methods by which one can judge the applicability of the model. And one of the method is leverage approach, which is based on the hat matrix. And there is some defined the value of the that is theoretical value of leverage, and above which value the a particular compound may be considered as the outlier or outside the domain of the model. Now there is a debate between internal and external validation, which is the better, but actually both should be applied. Internal validation can be used to check the robustness of the model, and external validation is used to show the applicability of the model for the new set of data. So external validation is considered of the gold standard and is the final one to judge predictability of the model. Now we'll see this. So these were some introductory slides about QSR model generation and validation. And from these slides, one can have an idea about the basic steps of QSR model development and how one can validate a model and judge the quality of the model using different metrics in a statistical approach. Next, I'll show some of the tools that have been developed in our laboratory to check the quality of the model and also to develop the model. These tools are available in these two sites, and one can use these tools free of charge by downloading them. These are all Java-based programs, so one need to have Java installed in one's computer. So these are some of the basic tools: the MLR validation, dataset division, external validation class, best subset selection, genetic algorithm, bias variance estimate. These are the small modules which are useful for different steps of QSR model. But here I'll focus on some specific tools like AD using standardization approach. So this is a method for determination of applicability domain of QSR model. So as you know, a QSR model is a combination of different descriptors, and the descriptor values of the training set, we can consider that they will have a normal distribution pattern. And for a normal distribution pattern, you know, the mean plus minus 3 times standard deviation covers 99.9% of the total population. So anything is which is beyond that range can be considered as an outlier. So based on that concept, one can standardize the descriptor values of the training and test set compounds using this expression, that the particular descriptor value minus mean of that particular descriptor value for training set compounds divided by standard deviation. So you get the standardized value. If the standardized value is less than 3, the maximum standardized value, considering all the descriptors, then one can be sure that all descriptor values, standardized values, are less than 3. So the model, the particular query compound will be inside the domain. Now one can also compute the minimum one. If the maximum one is more than 3, then one should also consider the minimum one. If the minimum one is also more than 3, then one can be sure that that particular query compound is outside domain. But if the maximum value is above 3 and minimum value is below 3, then one has to consider whether the particular query compound is within domain or outside domain. And in that case, one can use this expression based on Z distribution pattern. And here one can compute this S nu. If it is more than 3, then the particular query component will be outside domain, and if it is less than 3, then it can be within the domain. Next one is double cross validation. Is a method to check the quality of a model using the additional steps of validation. So this was published in Geometrics and Intelligent Laboratory Systems in 2016. So what is double cross validation? Here in QSR model development, as I've already mentioned, what we do, we divide the data set into 2 parts. One is training and another is test. And training set is used for model development, and the developed model is used for prediction of the test set compounds. But here, in case of double cross validation, we stratify training set compounds also, and we apply k-fold cross validation, where we get k calibration sets and k validation sets. And from these k calibration sets, we can derive k number of models, and we can apply this k number of models on k number of validation sets. And we can then check the performance of the model on the k number of validation sets, and we can select the optimal model based on different criteria, which can be based on model with the least mean absolute error of the validation set, or from consensus predictions from the top model. Consensus means when we are using more than one model. So that is a very good method of predictions where instead of using individual model, one can use more than one number of models, or finding best descriptor combination from the top one. Now the process of double cross validation can remove the bias in descriptor selection, because if we do not perform this, if we just use training set compound and develop the model, then we can have a biased selection of descriptors. Especially for small data sets, we can have a biased selection of descriptors, because for a fixed composition of training set, we can have a fixed composition of descriptor based on the strategy of selection of the descriptor set. But if we are applying the double cross validation, we can have opportunity to check more number of descriptor combinations, and the bias in descriptor selection is removed. Then intelligent consensus prediction, as I've already mentioned, that the consensus model is better than individual models, and usually for consensus predictions, the average of predictions from individual models is used. But in case of intelligent consensus prediction, which we have published in Journal of Cheometrics in 2018, we apply some strategies where we can use different models in an intelligent fashion. So what are these? There are three strategies, CM1, CM2, and CM3, and where we introduce the concept of qualified models. So qualified model means a model which is applicable for a particular query compound. So not that all models are applicable for a particular query compound. And to know that, we take the query compound and we select 10 most close training compound based on the chemical similarity, and we also check how far these close 10 compounds from the original query compound. And based on some predetermined threshold, if we find that we cannot get more than 3 close training compound for a particular query compound, then we can consider that particular model is not suitable for prediction for that particular query compound, because in the training set there are not sufficient number of compounds similar to that particular query compound. So the that model which has been trained on that particular combination of training compounds will fail to predict that particular query compound. So in that way, we consider only the qualified models, and then we take average of the prediction from the qualified models only. We do not use prediction from all the available models. The next method is weighted average prediction from qualified model. As I've mentioned that different models can perform in different ways for a particular query compound. So what we do, we again check the performance of the different models for the 10 close compounds in the training set for a particular query compound, and we check the leave one out predictions. And from the cross-validated prediction errors, we can actually penalize different models, and we can take weighted average predictions from different models for a particular query compound. So this is the second method. And in the third method, we select the model in a query compound specific way. So for query compound 1, maybe say model 3 is used, and query compound 2, maybe model 5 is used, and in that way we select our model for each query compound separately, and then we can predict their corresponding responses. And we can use also different criteria like Euclidean distance cutoff, applicability domain, Dixon Q-test, etc. And finally, we get the predictions from different consensus predictions, CM1, CM2, and CM3. And these predictions can then be compared with the performance of the individual models. And we have seen in most of the cases, the consensus models are better than individual models, and specifically CM2 and CM3 can win over the individual models. So this next one, this is prediction reliability indicator. So when we are using any model and doing predictions, so we get some values, but we need to know what is the quality of predictions. So we have applied the method for mainly MLR and PLS models to check the quality of prediction, which was published in ACS Omega in 2018. So here we are using three different criteria. One is based on the mean absolute error based leave one out predictions of closest 10 training compounds for a particular query compound. That one. Second criterion is based on applicability domain, and third criteria is based on scoring based on the proximity to the observed response mean, because it has been seen that the data points which are close to training set mean can be handled well by a particular model. So based on these three criteria, we can give the score to individual models, and then we can compare that is we can derive the composite score based on different weightage values. And finally, we can optimize these weighted values, and we can have our reliability of prediction from the composite scores. So it has been seen that 0.50, 0.5, this ratio is optimum in many of the data sets. Okay, finally, I discuss about the problem of the small data sets. In case the data set is very small, in many cases, we do not have sufficient number of data. We have limited number of data for model generation. In that case, it becomes difficult how to divide the data set into training and test, and to develop the model and then to validate. So we need to give special attention in those cases. So if the data set is small, a significant amount of information is lost due to the held out samples. Moreover, there may be a bias in descriptor selection due to fixed composition of training set. So how we can handle these problems? There are two methods. One is we can use double correlation approach, which actually eliminates the bias in descriptor selection by using two different loops, inner loop and outer loop. Actually more number of descriptors, more information, because for consensus models we have more the more reliability and more coverage of the applicability domain. And also one can give sufficient attention to curate the data set to remove the common error. So there should be a curation process, because the structure that is being used for normalization may be wrong. The normalization of bond may be incomplete. Some duplicates may be present in the data set. Other possibilities may be wrong charge, presence of explicit hydrogens in the hydrogen depleted structure, incorrect bonds, etc. So for that, we have a NIME workflow for for curation. So where we can use different nodes, like say element filters to remove the organometallics. We can remove mixtures by connectivity node. Then we can use RTKit's salt stripper, because in QSL you usually use the acidic or basic form. We do not use the salt form. Then we can also optimize geometries, we can normalize structure, etc. So finally, we get the curated data set. The small data set, which is chemically curated, then can be subjected to small data set curation, where we can apply duplicate analysis, structural outlier removal, response outlier removal, and activity cliff analysis. Duplicate analysis means one may have two different compounds, that means two different entries which are actually same compound. But it may so happen that their biological activity values reported to be different. So in that case, if they are sufficiently different, then one should omit both of them, unless you know which one is the exact one or correct one. Otherwise, they are close to each other, we can take average. Then we can use response outlier removal based on this mean plus minus k times standard deviation approach, where k is 3. So the biological activity value of a compound which is sufficiently different from rest of the compounds can act as influential observation. Structural outlier. If a compound is structurally very dissimilar from rest of the compound from the data set. Then activity cliff. Activity cliff means two compounds are very close in their chemical structure, but their response values or activity values are so close, so different that they cannot be handled by the model. So in such cases, activity cliffs should be omitted. And for doing so, we have some strategies which are based on the number of compounds which are actually showing the activity cliff behavior. And based on that number, we have different criterion which we can apply for addressing the problem. So I cannot discuss it in detail and may not be, it is very specific. So those who are not of this background, it may be difficult to understand. So I'm just skipping those. Now, in case of small data set modeling, starting from the curated data set. Say we have a number of curated compounds, and what we can do, we can use this double cross validation. So n number of compounds we can keep in each validation set. So we can generate a case number of K-star number of calibration sets and K-star number of validation sets. So this can be actually a very big number actually. And then we can apply our variable selection tool, maybe the genetic method or some other method like stepwise or so on. And we can generate model based on MLR or PLS. So we'll get the K-star number of MLR and PLS model. And then we can select the best model from the application of these models on the validation sets based on the MAE value for the validation set, that is least MAE value for the validation set or least MAE from the modeling set, or highest cross validation statistics, and some other method like some pooling technique and consensus approaches. So this small dataset modeler implements exhaustive double cross validation. Here actually huge number of iterations are done. So we are getting several number of combinations, and from that we are generating our models. And we are checking at each iteration the quality of the model using our usual metrics, some of which I've already discussed. So these are graphical user interface of the software tools that we have hosted in our website for small dataset curator and small dataset modeler. And this particular work was published in Journal of Chemical Information and Modeling in 2019. And recently we have launched this one, DTC QSR, which is a complete QSR package where the included features are user-friendly graphical user interface. It can develop both regression and classification-based models. All steps in QSR model development and validation are performed using the same software. Variable selection methods that have been used, genetic algorithm and best subset selection. Modeling techniques are MLR, LDA, and random forest. One can also determine activity domain, perform wide randomization. There are some graphs and plots generated, residual plot, etc. And one can do screening and prediction using this software. So in case of some problem in accessing the software or running the program, one can use this page, the frequently asked questions, or one can submit the query using the Google form given here. So these are the site addresses which one can access freely without any charge. So these are the website addresses from where different modules for development of QSL models and validations are available. So we have got funding from different sources at different points of time. For the beginners, if you want to know about QSR from the basic level, one can consult this book, Understanding the Basics of QSR for Application in Pharmaceutical Science and Risk Assessment. And one smaller version, Springer Brief, is also available, a primer on QSR, QSPR modeling, and some reference texts are available here. QSR in drug design, predictive toxicology, and advances in QSR modeling. And some more specific application of QSR are given here. In case of Alzheimer's disease, multi-target drug design, in silico drug design, and ecotoxicological modeling. And coming in 2021, in silico modeling of drugs against coronaviruses and application of chemometrics and cheminformatics in aquatic toxicology. So those who are interested, they can follow these books. So if you have any question, I don't know whether there is any opportunity to take questions or not here. You can ask. Thank you.
Thank you. Thank you, sir. Thank you for your excellent presentation, sir. Now, I would like to request Professor Jayanto Chattopadhyay to sum up the presentation, and after that, we will start the question answer session.
Good evening, everybody. Thank you, Professor Roy, for such a difficult presentation. I am, quite blunt about it because I studied. I had the opportunity to study medicinal chemistry 40 years ago. So in Jadavpur University in those days, I had the opportunity to do a little bit of QSAR work under Professor Dey. And due to that thing only, today Dr. Hossain and Dr. Professor Punarvesh Chakraborty has asked me to summarize all this. So this is similar to, asking a bishop, to host a MTV session. I am completely out of the drug discovery world. I learned QSAR 40 years ago, but then I can relate what I learned in those days and what is happening now. This is 2021, and I am talking about 1980. Now, Professor Roy started with my familiar equation that biological activity is a function of physicochemical activities or structures of compounds. And during those days, we learned from Corinne Hunch that there can be a equation can be developed between biological activity and the physicochemical parameters of potential drugs. And later on, in those days, we used to determine partition coefficient of a few molecules in the laboratory and find out some physicochemical parameters from the literature, and some are experimental partition coefficients between nocturnal and water. And then we tried to correlate between published biological activity and the physicochemical parameters of a 5-6 compounds that we nowadays learn that we call it a small set of compounds, and try to find out a statistical relationship using Hans formulation. But that was 40 years ago. And after that, from Professor Roy, we have learned that this quantitative structure-activity relationship has become an extremely sophisticated statistical technique, as you understand today. And the subject matter is so technical as on today that without a good course on statistics, it's extremely difficult to understand what is going on there. Such is the complexity of the modeling, but I will try, in my own way, to explain people like me what is going on now and what I understood from there. Maybe my understanding is different from the reality. So what happened, what Professor told us, that the data set of a chemical compound, usually the QSAR, you have to build a model, and for model development, there are feature selection using molecular descriptors. Now these descriptors are, some kind of, in my belief, there are physicochemical parameters. Then there are, 2D descriptors, 3D descriptors for some three-dimensional property. And in earlier days, we used to have a kind of Hammett constant, then partition coefficient, then electron density on a particular atom. These are the kind of descriptors we used to calculate. Professor, they used to calculate by quantum mechanical methods, the charge density on different parts of the molecule. And we tried to kind of correlate with a series of compounds and find out a relationship, by using regression coefficient. And, in that case, you have to, kind of solve a series of differential equations, kind of The relationship could be really linear, the relationship could be non-linear. But then again, the subject has become so sophisticated in statistically, at least nowadays, that the model building, you have a training set of compounds. The compounds whose biological activity is known and their physical features are also known. So using the training set of compounds, we build a model. And after these equations are established, then we test it for a test set of test data. Now, once it can predict after the training set, the testing of the model happens, and what we call the validation of the model. And the validation of the model, there are lots of ways of validating the model that we have understood from Professor Roy. And then there is a question of that user is not only applicable to drug design. There are other fields, particularly for predicting toxicity. So design of safer chemicals and green chemistry is nowadays a big area. So for designing safer chemicals and predicting toxicity, we understood that same principles of the contribution of different descriptors in the toxicity, in the toxic manifestation. That is also a biological activity, of course. Is another interesting area where QSAR can be applied, and by that probably safer molecules, less toxic molecules can be designed for any application. Coming to basic utility of QSAR in drug design, in the drug discovery and research, he has given a number of examples of success stories of QSAR. One molecule I remember now, norfloxacin, and there are certain specific molecules he mentioned which are the outcome of quantitative structure activity relationship. So the initial days, he discussed a lot about the validation of QSAR models, and also he talked about some OECD guidance for QSAR model development. I remember in our times there was a kind of some kind of criticism happened on the QSAR researchers. In those days, when there are people who used to do some theoretical research from past data and used to predict some, predict some outcomes or biological activity based on the past data, and they used to publish a paper. So in those days, there is a idea that if you really make a model and try to predict something, do some experimental work and in a set of compounds, and then and find out your results experimentally. But that was 40 years ago I am talking about. After that, the model building and the the validation of the models has become tremendously sophisticated. And you understand, there are a lot of researchers, statisticians probably worked on this, and question of outlier removal and curating data set, that is also he mentioned. Correcting the problems of QSAR module from small data set. Naturally, if you have a small data set, your naturally it is not a big large data, so chances of error will be there. So how to curate small data set? There are a lot of issues like data homogeneity, errors in descriptor selection, collinear descriptors, unacknowledged omitting data points. Supposing I have a molecule and I find that if I include that molecule, my statistical correlation falls. So I remove that molecule from that data set and my correlation improves. So this is a bad practice that this OEET guidance says. That is what I understood. So validation of QDAR models, and particularly in small data set, what I have understood from him that there can be a bias in descriptor selection because data set is small. So then there are kind of lot of validation models. He talked about intelligent consensus prediction model, and particularly selection of data set selection. He talked about that, and he talked about internal and external validation model. Once again, trying to grasp from all these difficult statistics and jargons from almost layman's perspective, I would say that the purpose of discovering drugs, at least QSAR is becoming highly statistical and mathematical. So naturally, uh, the books he suggested, some of his books he suggested for anybody. I know students are not here, but any young faculty who is planning to enter QSAR area, he has rightfully said that initially we have to go through his books to understand what is going on today, equip ourselves, and I would suggest them to go through the books of basic statistics, because QSAR is so full of statistics. So naturally, unless you have a good understanding of statistics, it is pointless to enter into the QSAR area. And then he talked about his job in chemometrics, drug theoretics, and chemoinformatics laboratory in Jadavpur. In his commercial days, many softwares are freely available from his website. This is a very important news for those who are planning to do work in QSAR area. He has made available lot of important softwares, free of cost. So this is a great service. Those who are interested in QSAR work, they can access those websites and get the help of those softwares. So this I would congratulate Professor Rai, because it is a big help to those who will be working in young people who will be working in QSAR area, and those who do not have the funding to buy sophisticated softwares. Finally, I would say that end of the day, discovering how we are in the the mission of pharmacy is to discover important, effective low toxicity drugs against a host of diseases that is happening. There are different people from different domain are trying. There can be a biochemical rationale. Somebody can find out from the mechanism of action, how the virus sits on the cell surface, the enzymes and all this. There are a lot of modeling, and there are a lot of there are other ways of doing that. Docking, molecular docking, if you know the structure of the protein, lot of protein structure are elucidated, and there are a lot of docking work going on, and based on that people are getting the leads. So but end of the day, I would say that when people discuss about drug discovery, or even in lay press people discuss about drug discovery. During corona time it happened. A lot of people talked about statistics, how when the corona epidemic will end and all. So I had a statistics professor as my friend. So I said that statistics is all meaningless. There are lots of models, but very few models can predict the real thing. We really do not know when the end point will come. And there are a lot of discussions on herd immunity and all, but everybody is and there are a lot of experimental research came that BCG vaccine is effective, or that could be very effective in corona. But there are a lot of statistics involved, and there are a lot of misinterpretation of statistics. At one point of time, Professor Ray mentioned about this, that misinterpretation of statistics is another critical area. He talked about double cross validation and all sophisticated things. Misuse and misinterpretation of statistical data, because normally those who are not highly trained in statistics, they will apply their common sense for finding out an outlier and all this. But in statistical world, you have to only come up with real statistic and data and the statistical conclusions. Once again, it is a specialist subject. So with all this, I am very note what was started in Jadavpur University 40 years ago, Professor Dev, started working in QSAR. Now that has probably reached pinnacle, and people like Professor Roy is now heading that initiative. So I am very proud of that university of my alma mater, that so much good work is happening. So I am once again thanking Professor Roy for such a elaborative, of course, highly technical lecture. Thank you very much, Professor Roy.
Thank you very much, Professor Chattopadhyay. You have nicely summarized the topic. Just I will add chemistry, because in drug discovery field, mostly the structure-based approaches are more used, because now we know the receptor structure and all. So we use structure-based approach, fragment-based approach more than QSR in drug discovery. That is one point. And but QSR is being used in predictive toxicology and regulatory toxicology to a great extent nowadays. So main focus of QSR is nowadays is in regulatory aspects. And another point I will mention that all models are wrong, but some of them are useful.
Absolutely, absolutely. This is what they talked about the corona epidemiology statistical models, that there are many models out there, but very few are really useful. Is it what you are saying? That? Okay. Thank you very much.
Thank you, Professor Chattopadhyay, for nicely summing up the presentation of Professor Roy. Professor Roy, I would like to ask you a couple of questions. Just I'm curious to know, as I have very little knowledge about QSR. As for the drug discovery aspect, the determination of IC50 is very important for measurement of efficacy and potency of the drug. So how QSR makes acceptable and lower the IC50 of a new molecule during the design by QSR?
You repeat the last part of your question?
How it makes an acceptable and lower the IC50 of new molecule by QSR model? Lowering the IC50, it increases the efficacy and potency of the drug. Is it possible to make to lower the IC50 by the QSR modeling?
You mean to say, okay, so when IC50 reduces means that actually activity increases.
Yes,
so that means you want to design a compound with higher activity, and that is the purpose of QSR. Because when you are deriving a model, the model has several features in the form of descriptors in it. So that will guide you to design new compound in a systematic fashion, so that you can incorporate features which increase the activity, and eliminate the features which are not required for that. And in that fashion, you can design a compound with higher activity or lower IC50 value, I would say, and you can have the predicted higher PIC50 or lower IC50 value. And that is possible, and then you can actually synthesize that compound and check whether that is working or not. So that is the purpose of QSR.
Yeah. So is there any specific data, like how much fold decrease of the particular IC50 that will make significant activity or potency? How much decrease of fold?
Quality of the chance to become good, and then it will have potential to give good quality of prediction. So it is in terms of statistics only, it's the quality of prediction. So how good or bad you can predict. This is all about prediction.
Okay, sir. Sir, another question: is it possible to perform the 3D QSR study by the inhibitors obtained sample of inhibitors obtained from different experimental study? Like in QSR, I think data are studied from single experimental study data set. If data obtained from different experimental study, is it possible to study the 3D QSR study?
If you have your derived data from different experiments, you cannot do a regression-based QSR, but you can use that for classification-based QSR.
Okay, sir. So my last question is, as we are involved in, as our always we think about the cell culture study, animal study. We are always limited in this experimental study, biological activity in cell or animal model. So, what is your experience like when a pharmacore or lead compound is designed by QSR study, and what is the difference of the variation of data obtained from QSR and effect obtained in the cell culture and the animal model?
So see, if you have the in vitro data, that only will give you the receptor binding affinity or enzyme inhibition or like that. But if you do not have the in vivo data, you have the pharmacokinetic treatment. So, hello.
Hello, due to the network issue, I think it is
Hello.
disrupting. Yes, sir. Yes, sir. It is audible.
Yeah.
So my question is, during the ADMET study, like adsorption, distribution, like toxicity study obtained by the QSR, when you have obtained some data or some some predictable data, toxicity, related toxicity, or maybe from environment hazards, when it is sent to the lab for wet lab study, what is the variation of response? That is my question. Like toxicity obtained from the new molecule design. Toxicity obtained from.
You mean to say the predicted data from the model and the actual response? What is the variation that you want to say?
Yes. In terms of efficacy or toxicity.
High. So that depends on the quality of the model. That depends on the quality of the model, and that can vary. And that can vary from compound to compound also, because for some structural features or some structural category, the prediction may be of good quality. For some other category, prediction may be of a different category. So it is specific to the chemical class, and also depends on the chemical similarity to the training set compound.
Okay, sir. Thank you, sir. A question asked by Mr. Devanjan Sen. He asks that in computer-aided drug design, after we use Lipinski rule of 5 in order to analyze the drug ability of a ligand, but Lipinski in this original work published in 1998 says the oral absorption of the drug depends upon the dose. What he has written, fifth rule, that we consider it's an essential criterion for virtual screening SBDD based heat identification method. But he doesn't. There is no question actually. In Q&A box, you can see, sir, his question from Devanjan Sen. In
Lipinski's rule of 5 is used mainly for the orally used drugs to understand the bioavailability of the compounds. So whether they will be absorbed or not, etc. But so that is used as a common filter while screening. But what is the question? I didn't understand.
Yeah, question actually he didn't ask any question. Okay, next question. What is the difference between the QSR and QSPR?
QSPR?
Yeah.
So when the response is property, then it's called QSPR. Both are same. The concept is same. When you are using activity as the response, then it's called QSPR. And when you are modeling some property, say for example, if you are modeling log pairs, partition groups, then it will be QSPR.
Thank you, sir. Now I would hand over to Professor Pranabes Chakraborty, sir.
Thank you, and warm, hearty wishes to my younger brother and Professor Kunal Roy for his exclusive and unique deliberation in front of our faculty members.
Thank you very much. Sir, I would just like to know about the participants. They are from different colleges under Macaut, or?
No, actually, participants are not only the colleges from Macaut, some other universities, from some other universities also, they have joined from different parts of India. So
Okay.
I'm really thankful to Professor Kunal for his extensive, exclusive, stepwise analysis of QSER, and we will hope and expect from the university and the Department of Pharmaceutical Science and Technology that in future, when the physical mode will start, as today he has helped us, in the near future, hope for the best, will come together physically, and at that time we will request him to join hands with us so that our students can get more idea and conception about the subject.
But
Because
I am more comfortable online. Otherwise, I have to travel to Waringa.
no, well, we can arrange it in Salt Lake. We can arrange it in Salt Lake. Yeah.
Okay.
So really, I'm very happy, and I hope and expect that our learned faculty members, directors, principals, professors from different colleges and universities, they have enjoyed the lecture and they have learned. And I'll request, as Professor Chatterjee has requested, to go through the books of Dr. Kunal Roy, because he is one of the legendary figure in this QSER, computer-aided drug design, and hope that you will certainly learn what you want, and you will develop your conception from the perceptual level. And simultaneously, I'm congratulating Professor Chatterjee for his nice and unique summarization of the total deliberation. With this, once again, I am extending my hearty, warm wishes to Professor Rai, Professor Chatterjee, Professor Hussain, and our all-time friend, Mr. Prem Kumar, who is always behind the cloud and helping from the early morning to midnight for the success of our FDP program. And with that, once again, I am extending my wishes to all my learned faculty members. Thank you all. Over to Professor
Thank you
Hussain.
very much. Thank you very much.
Thank you, sir. Thank you for your closing remark. I would like to thanks to all dignified panelists today and our eminent speaker, Professor Rai. Thank you, sir. And I would like to thanks our dear participants, learned faculty member, research scholar, and industry person for patience hearing. And tomorrow onwards, 7 p.m. onwards, we will listen lecture of technical session 5 from. We'll listen from Mr. Lolit Kumar Pradhan. He is person.
No. Tomorrow lectures are there.
From 6 p.m. sir.
From 6
technical
p.m.
session 1.
Mr. Arjun Gothakurata.
Oh. There is not Arjun Gothakurata. Yeah.
Tell that their topic also.
Yeah, I apologize for this. In day 5, at 6 p.m. our technical section 5 will start. The speaker will be Mr. Arjun Gothakurata. He is also a stalwart person from industry background. His topic will be sterilization product tracking, business process automation, and automatic program tool. And from 7:30 p.m. onwards, we will listen talk from Mr. Lolit Kumar Pradhan. So tomorrow we'll meet again from 6 p.m. Thank you.
And lastly, I request Professor Kunal Roy to join on Sunday.